and welcome to today's From the Journal session, a new event type at the AAAS annual meeting, where we invite authors from recently published Science Family Journal articles to discuss their work and its implications. My name is Matt Wright, and I'm the Deputy Director of the Science Press Package. Today, we will be hearing from the lead authors of a closely related pair of research papers published on January 12, 2021 in Science Signaling. Together, the papers highlight how the SARS-CoV-2 virus may exploit newly identified sequences within cell receptors, known as short linear motifs or SLIMs, to enter and replicate within human cells. The results also hint that with further research, these sequences could serve as targets for new therapies to treat COVID-19. Here to discuss the results of this research today are Bálint Mészáros, a senior postdoctoral researcher at the European Molecular Biology Laboratory in Heidelberg, Germany, and lead author of the first paper, Short Linear Motif Candidates in the Cell Entry System Used by SARS-CoV-2 and Their Potential Therapeutic Implications, and Johanna Kliche, a doctoral student in the chemistry department at Uppsala University in Sweden, and lead author of the second paper, Cytoplasmic Short Linear Motifs in ACE2 and integrin beta-3 link SARS-CoV-2 host cell receptors to mediators of endocytosis and autophagy. They will overview the motivations for their work, the findings, and their implications. I will be asking questions of the speakers after each of their remarks respectively. The speakers will also be available to take your questions in a follow-up Zoom room hosted by the AAAS annual meeting at 2 p.m. Eastern Time on Monday, February 8th. More information about how to access that room is provided in the annual meeting entry for this session. Without further ado, I introduce our first speaker, Bálint Mészáros. Bálint?
Thank you, Matt. It's really a pleasure to be here. And I'm going to be talking about very collaborative work. Truth be told, I'm only going to be talking about the first part, which is the model building and the computational part. And my colleague Johanna is going to talk about the experimental follow-up that we did. So on this first slide, we have a very brief overview of the life cycle of the SARS-CoV-2 virus, how it attaches to the host cell, which is in this case a human cell, how it gets inside, replicates, reassembles, and then leaves the cell to infect other cells. So what you can see on the right is pretty much one of the most fascinating properties of viruses, at least for me. Even the deadliest viruses like Zika or Ebola typically only contain a handful of proteins or maybe a few dozen. But in contrast, a human cell contains more than 20,000 different proteins. And this discrepancy in size is apparent in the physical realm as well. So on the lower right, what you see is a recently published image of a single human cell being infected by SARS-CoV-2, which is small red dots. And you can see there's a huge difference in size between the virus and the human cell. So this means that viruses are very small and very compact. So in a way, when we think about them, we don't really think of them as separate living entities, but as master hijackers of the cell. So they, instead of doing everything that they need in order to replicate and infect cells on their own, they typically subvert the processes that are already there in the human cell in order to do this. And during my talk, I'm going to be focusing on how they do this by concentrating on the first step, which is called the viral attachment. So we had this fairly early model early on when the whole pandemic started, because SARS and MERS operate in a very similar way. So we know that the primary attachment between the virus and the host cell, the human cell, is mediated by a protein called spike on the virus surface, which can bind to a human protein on the cell surface. And this protein is called ACE2, which is an enzyme and can regulate the blood pressure. So this is what we knew, and this is a very clear and straightforward picture of how the virus gets attached to human cells. But in fact, biology is very seldom clear and straightforward. So even looking at this very early model, it raised two questions for us. So first of all, why does it attach to ACE2? ACE2 is a blood regulatory enzyme, and messing with blood pressure in humans doesn't seem to have a clear benefit for the virus. And the other question is, how can this virus infect tissues like the lung tissues, which virtually have no ACE2 in them? So we are going to tackle these two questions by looking at these receptors or ACE2 and other receptors more closely, and we're going to focus on the intracellular tail. So what exactly do we know about ACE2 as a protein? We know that it's fairly large. The outside part, which is sticking out of the cell, has over 700 residues. That is what carries the enzymatic function, which participates in the regulation of blood pressure. but it also has a fairly short bit that is sticking inside the cell called the intracellular tail. So this intracellular tail is composed of 44 residues only. And if us, like structural biologists, look at this sequence composition, it becomes fairly evident that it has a lot of charge in it, so charged residues and residues that can be chemically modified to change their properties. And these type of sequence properties all points towards this being a very flexible so-called disorder protein, which can probably interact with a lot of other proteins inside the cell. And these type of proteins are very, very common in cell signaling and regulation. So in these cases, when pretty much the only information we have is a protein sequence and nothing else, we very often start looking at evolutionary signals. So here we collected ACE2 sequences from different organisms, ranging from humans to hagfish. And hagfish are fairly primitive vertebrate fish, which have diverged in evolution from humans about 500 million years ago. And still, although they diverged such a long time ago, the ACE2 protein, the actual function inside the cell, is pretty much the same in all of these animals. But the sequence isn't, because proteins slowly gather mutations throughout evolution. And most of these mutations are harmless and don't really affect the function. So we can harness this information by looking at places in the protein sequence which are very well conserved in evolution, like these colored patches that I marked on these sequences. These are the areas of this protein, of this intracellular tail, which seem to be very robust throughout evolution and don't really change that much. And whenever we see these little patches, it typically, these conservation patterns typically happen because these are functional regions inside the protein, so they do something. Question is what? So when we see these little islands of conservation inside disorder proteins, we typically think these are so-called short linear motifs. So see, these are little patches inside a sequence which can interact with other proteins and mediate some kind of function. For example, if you look at the middle one, the blue one, this specific motif is called ApoPTB. The question is, if it interacts with something, what is the partner and what does it do in the cell? So based on our resources that we have built around these short linear motifs, we actually have a server we've been developing for 15 or 20 years in our group, where we collect the function and the interaction partner of these short linear motifs. And based on this, we can come up with a prediction. What are the molecular interaction partners of ACE2R? And you can see these three colored boxes. These are three patches that I showed earlier. But in reality, the first box is actually a bit more complicated because it's not a single short linear motif. It's actually 4 different motifs overlapping each other. And there is a modifiable residue, a tyrosine inside of them, which can regulate which interaction is actually active. So it's like a molecular switch, which Johanna is going to talk about in a bit more detail. But for now, my point is that if you know what the function of these short linear motifs, then you can come up with a model of what the molecular interaction partners are for ACE2. And based on that, you can have a guess of what kind of pathways these interactions belong to. And if we do this type of analysis, we can see that most of these interactions actually are connected to something called endocytosis, which is the process through which the cell takes up material from the outside. And endocytosis is very critically important for a lot of viral processes. So in light of this, what we are proposing is that ACE2 is actually much more than an enzyme involved in blood pressure regulation. And the way to understand it is to look at the inside the cell and to try to untangle these interactions. Now, this gives us a fair idea about why ACE2, but it doesn't really explain how can this virus infect tissues that have no ACE2 at all. So for the second question, again, we are sticking with short linear motifs. So if you look at the sequence of the spike protein itself, we can see that it also contains a short linear motif called RGD, which is composed of these 3 residues next to each other. And what we know about RGD motifs in general, and this has been known for 20+ years, is that RGD motifs can, under certain circumstances, interact with a protein family called integrins. So integrins are, again, cell surface receptors, similarly to ACE2, although they have very different functions in the cell. Now, the fact that the spike protein has these three residues isn't proof. It's very, very circumstantial because these three residues can happen next to each other just by random chance. So how can we decide if this is actually a functional interaction site for integrins or not? So actually, we know a fair bit about integrins because they are connected to cancer and autoimmune diseases. So we know that these are involved in cell attachment. We know that a lot of known viruses actually hijack integrins and use them to get inside a cell, viruses like HIV or HPV. We also know that integrins can regulate endocytosis. And we also know that under normal circumstances, integrins and ACE2 interact on the cell surface. So they regulate each other. So just like we did for ACE2, we can look at the intracellular tail of integrins to get an idea what might be the molecular connection. So if you do that, and again, we are looking through hundreds of millions of years of evolution, we can see these short linear motifs. But integrins are fundamentally different from ACE2 because integrins have been very well studied. And actually, there were 3 different linear motifs that have already been identified. These are the red and the blue ones. So the 2 blue ones are actually the same that we just discovered in ACE2. And these are connected to endocytosis, which again strengthens the idea that integrins are co-receptors because they can mediate very similar signals as ACE2 can. But we weren't content with this explanation, so we started looking even more. And we discovered a new short linear motif, which is marked in green, which is called Lyr. And the definition and the name of this linear motif isn't very revealing, but Lyr motifs are actually known to be very intimately connected to autophagy. And we actually found the same Lyr motif for a very similar Lyr motif in ACE2 as well. And we also know that autophagy, which is the process where the cell digests certain components of itself, is actually hijacked by a lot of other viruses. So based on this information, we can further complete the picture that we have about how this viral attachment works. So apart from ACE2, the spike protein can probably interact with integrins and propagate signals inside the cell, which again are connected to endocytosis and autophagy, the 2 key processes which are very advantageous for the protein or viral infection. All right. So in light of this, it's a fuller picture that emerges, which says that SARS-CoV-2 uses highly redundant parallel mechanisms to invade the human cells. And in reality, the picture is even a bit more complicated because the cell surface, the human cell surface, contains other proteins called proteases, which have the primary role of activating other proteins. And integrins have a type of protease that activates them called furans, and ACE2 has another type of protease called TMPRSS2, which activates them. Now, it turned out fairly early on that the spike protein actually evolved these activation sites, which can use these 2 proteases as well. So it doesn't only hijack receptors, it also hijacks the activating proteases. And to make it more interesting, actually, when one of these activation happens on the spike protein, that creates yet another short linear motif called C and R. And the function of this motif is to be able to interact with a 3rd receptor called neuropilin. So this was actually published in November in Science in 2 back-to-back papers where they did a lot of in vitro and cell-based assays to prove this. So knowing about these 3 receptors actually can give a better indication or is more in line of the knowledge of the tropism of the virus that we have. So what tissues does it infect? If you look at the expression patterns of integrin beta-3 and neuropilin, you can see that these are very well expressed in lung tissues and other tissues. So one of the key reasons why a virus would evolve separate pathways, separate parallel pathways, hijacking different receptors is first of all to get a more robust infection, to be able to infect different types of tissues. And this type of knowledge has to be taken into account when we are developing drugs. So on the right side, what you can see is a couple of suggestions that we made understanding these processes. And this approach is called host-directed therapies. So in this scenario, we are not targeting the proteins of the virus itself. We are targeting these processes which are hijacked by the virus by attaching to cell surface receptors. So we have three categories. We have compounds that can counteract the cell attachment itself. We have compounds that can shut down endocytosis. And we have other compounds that can modulate autophagy, so the key cellular pathways that are at play. And keep in mind that most of these compounds, so these compounds already exist. And a lot of these compounds already have FDA approval. So it would be very easy to repurpose them and to test them at a clinical setup. Now, there is no guarantee that these are actually going to be good antiviral drugs, but it's very easy to test. And these are very good candidates to counteract what the virus does to the cell. So in closing, I would like to thank my co-authors and my collaborators, and I would like to thank you for your attention.
Thank you, Bálint. I had a few questions for you. I'll start with one. In the past year, we have seen a lot of research and associated news coverage that addresses the role of the ACE2 receptor in mediating SARS-CoV-2 infection. This pair of studies seems to indicate that we've been missing some important parts of the picture, especially regarding other receptors such as integrins and neuropilin-1. Is this a fair assessment, and why or why not?
I would say it is, at least in my point of view. This is a very particular story, and I think we can learn a lot from it. So when the whole pandemic started, a large portion of the scientific community directed their efforts to try to understand how this virus works and how it gets inside the cell and what it does inside the cell, which was an excellent call. Right? So a lot of labs started working 24/7 on this. And we already had two key pieces of information based on SARS and MERS that happened in the past 2 decades. First of all, ACE2 is the receptor for the virus. And the second is that lung tissues have a high level of ACE2. So based on this, it made complete sense to focus our efforts on ACE2. Now, the only problem is that one of these 2 key pieces of information, namely that there's a lot of ACE2 in the lungs, is false. And it's based on earlier papers, which turned out to be unfortunately not true. And now there are a lot of studies that say that ACE2 is very lowly expressed or not expressed at all in healthy lung tissues. And this is primarily a lung disease, or at least it starts as one. So I am not saying that everything that we've done on the ACE2 front is meaningless. It's immensely valuable, right? It's going to have a very prominent role in developing pharmaceuticals, because, for example, the intestinal infections or the heart infection very much depend on ACE2. So all of this focus will pay off in developing drugs, but I think it undoubtedly stole a little bit of focus on the fact that viruses typically don't use a single receptor. They use a combination of receptors. So people had very low incentive to go hunt for more, which actually I think it took so it led to this knowledge gap that we have, or we had. And I'm very pleased to see that more and more groups are focusing on finding other receptors as well. So it's becoming more balanced. And I think there's a lot to learn from this story.
Wonderful. That's a good lead into my next question. Given your work to characterize a large number of these SLIMs in these relevant receptors, what new research directions and questions might your findings prompt among the COVID-19 research community?
So I think our work is going to be useful in several ways. I think it has a conceptual message. I think if it only achieves that we emphasize that there are typically parallel receptors and parallel mechanisms that viruses exploit, then we would be happy. This is actually not a new concept. We knew this happens for other viruses as well, but it wasn't really emphasized for this virus. Hopefully it will help people to be more aware of this. Also, I think at least one of the key findings that I am most enthusiastic about is the link between autophagy and integrins, which is a very new link. And hopefully the scientific community will have an incentive to focus on this. And technically, I think there's an interesting aspect of our work, because at least what we do, all of the techniques that we use, like looking for sequence patterns, aligning sequences, doing structural modeling, actually it's not fully novel. And I know it sounds bad. The novelty of our results and our work doesn't rely on the techniques themselves, but the combination of these techniques. So you can build a very complex pipeline and get answers to very complex questions with very simple methods if you build them right and if you have an immense background knowledge of the biology, right? Anybody could have spotted these motif patterns, but there is a reason why they didn't. For example, my boss, Toby Gibson, has been doing this for over 30 years. So he just looks at a sequence and spot everything, or at least a lot of things that other people can't, right? So background knowledge and the creative combination of simple techniques can go a long way. So I encourage people to have a broader discussion between disciplines to initiate these complex approaches.
Great, thanks. Your work also breaks new ground by investigating previously overlooked virus propagating protein interactions within the cell after the virus has gained entry. How likely are these interactions to yield totally new drugs that are effective and safe to fight the virus? And might such intercellularly targeted therapies pose a higher risk of off-target effects by chance?
Well, that's an excellent question and a very, very difficult one. So first of all, if you look at it close up, we identified new interactions that are probably hijacked by the virus. So if you can block these interactions, these are definitely new targets that we could try. A problem is that these SLIM-mediated interactions are actually not that straightforward to inhibit. So to design small molecules to disrupt these interactions is far from trivial. But these are definitely new targets that, since we didn't know about these interactions' existence, these are definitely novel. Now, if you take one step back, these proteins that we identified as interacting partners of these receptors, in turn interact with other proteins, which interact with other proteins, and these constitute these pathways that lead to endocytosis and autophagy. So the interactions that we identified aren't the only points where we can intervene with these processes. And that's why we can think about drug repurposing, because a lot of drugs have been actually developed to shut down endocytosis or shut down autophagy or modulate autophagy. And these drugs should be tried out if they work in this setup. So in this sense, we are not the first to say that endocytosis and autophagy is important, but we are the first one to prove there is a molecular link between integrins, which are co-receptors, and autophagy. So we'd like to try to give it a bit more attention for people to try out the repurposing of these drugs. There are hundreds of thousands of compounds that have been developed. The trick is not to develop, in a lot of cases, not to develop new compounds, it's to figure out what the already existing compounds can do. So we are urging people to focus a little bit more on these processes and compounds that are connected to it, because we really do think there's a lot to explore there.
Wonderful, thank you, Bálint. I would now like to introduce our second speaker, Johanna Kliche. Johanna?
Thank you, Matt. I'm delighted to be here, and I'm going to present our paper now. So I'm going to start off with what Bálint left off with, and that is their final figure. So again, we can see the spike protein from SARS-CoV-2 and its known and putative host cell receptors in the human host cell membrane. And what Bálint and his team did was to look at the cytoplasmic tails of ACE2 and integrin beta. And they predicted a plethora of short linear motifs within the cytoplasmic tails. Additionally, they proposed a phosphorregulation of some of the binding motifs. So what my group and me have been doing is to functionally assess these motifs by affinity measurements using fluorescent polarization. And I've depicted here the motifs which we actually tested in red. So first, before we get started, I want to introduce the principle of fluorescent polarization. So you have a small molecule, in my case, that would be a peptide, to which a dye is attached, and you can excite the dye and it's emitting light. And because of the small molecule, it is tumbling in space so that the emitted light is depolarized. So when I add now my protein of interest and it's actually binding to the peptide, this results in a slowing down of the tumbling, so that this results in a polarization of the emitted light. So if I look at the saturation curve, so here I'm titrating my protein of interest against a constant concentration of FITC-labeled peptide, then if a binding event occurs, there's an increase in the FP signal. Whereas when I have a displacement, I have a preformed complex between the protein domain and my FITC-labeled peptide. And I'm titrating with a peptide which is not labeled. So if the unlabeled peptide is binding to the protein domain, this results in a displacement of the labeled peptide and thus a decrease in the FP signal. And we've been conducting both kinds of experiments. So to start off with, I'm showing here the C-terminus from ACE2, and there are 3 different types of peptides which we have tested. So first, the orange one to the right was predicted by Bálint and his group to contain a class 1 PDZ binding motif. And you can see the representative structure of one of the PDZ domains in the right bottom. So this is the one from SNX27, which is involved in the retrograde trafficking of membrane receptors. The second peptide which we have been looking at is the one depicted in green. And it was predicted to contain an overlapping motif for the mu2 subunit from AP2. And AP2, the AP2 complex is an adapter for clathrin. So it is part of the clathrin-mediated endocytosis. Whereas when the tyrosine is phosphorylated, Bálint and his group predicted that this will create a binding site for an SH2 domain. So here you can see in dark green a representative structure of FIN, which is part of the ZARC family of kinases. So first, to start off with the C-terminal PDZ binding motif. So here I titrated the protein domain against the FITC labeled ACE2 peptide. And I'm depicting here 2 of the binding curves of the PDZ domain which we found to bind to the peptide. So this is the one, the PDZ domain from NERV3 and the PDZ domain from SNX27. And for example, NERV3 has been shown to be co-localized with ACE2 in intestinal tissue, and both are involved in sodium-dependent transport. So we could validate the PDZ binding motifs. So the second peptide which we assessed here was the one that had the overlapping binding motif for the mu2 subunit and the SH2 domains. So in the upper panel, you can see displacement curves. And we could show that the mu2 domain from the AP2 complex binds to the unphosphorylated ACE2 peptide in a range of 100 micromolar. Whereas when the tyrosine is phosphorylated, this results in abolishment of the interaction. When we tested the SH2 domain from NIC1, we could not find any significant binding to either of the 2 peptides. This prompted Bálint to revisit their predictions, and so that they suggested that the ZARC family of kinases, or the SH2 domains from those kinases, bind to the peptide. So in the right bottom, you can see then the saturation curves for FIN SH2. And we could actually see that the phosphorylation of the tyrosine 781 enables the interaction with the domain, so that we can propose that there's a phosphorylation switch mediated by tyrosine 781, enabling either the binding of the mu2 subunit or of the SH2 domain. Secondly, we looked at the Lyr motif, which has been predicted in the integrin beta-3 tail. So again, in the left upper panel, you can see the peptide which we've tested. And to the right next to it, you can see the structure of one of the ATGA domains, which binds to the Lyr motif. If we look at it within the crystal structure to the left bottom, so here we can see the LC3A domain in complex with the Fuco1 Lyr motif. And they're the 2 hydrophobic or aromatic residues which dive into the binding pockets. And both upstream and downstream of the core hydrophobic motif, there are acidic residues which interact with the basic patches within the protein domain. So these are the red patches within the structure. And acidic residues can in certain circumstances mimic the phosphorylation status of a peptide or vice versa. And this is why we look kind of extensively at the phosphoregulation of this binding motif. So this would be the binding curves to the right bottom. And we could show that, that phosphorylation of amino acids upstream of the hydrophobic motif, so for example, that would be serine 778, can enable the interaction or promote the interaction. And interestingly, if there's concomitant phosphorylation of residues upstream and downstream, so the tyrosine and the threonine, then this there is a synergistic effect on the binding. So if I go back to the final figure from Bálint, and look at their predicted motifs, we could confirm that there is a PDZ binding motif in the ACE2 C-terminus. Additionally, we could show that the phosphorylation of the residue 781 can enable the binding of the SH2 domain and abolish the interaction with the mu2 domain from AP2. And interestingly, we also found the phosphoregulated Lyr motif in integrin beta-3, and Bálint has already said it, that has biological or could have biological relevance in a setting besides SARS-CoV-2 infection. And lastly, I want to state that what we've been doing here is the functional validation of the binding motifs, which does not equal biological relevance. So it still needs to be assessed by cell-based studies whether the receptors actually make use of their binding motifs. And that will then have implication for SARS-CoV-2 infection. And lastly, I want to thank the team. So my group and my co-authors, and our research has been funded by the Swedish Research Council, and then importantly of the people who actually took us on and made all these predictions. So Bálint and his team.
Wonderful. Thank you, Johanna. I have a few questions for you as well. And I'll start with, as you've explained very well, your work experimentally verified the interactions of several SLIMs that were identified by the study that Bálint described earlier. Looking at the SLIMs that you chose to investigate, it seems as though we may be hearing quite a bit more about integrin beta-3 in the near future. Is this fair to say? And are there other specific receptors to watch for in future research efforts, either from your group or others?
Yes. So I would say that receptors, by acting as linkers between the extracellular and intracellular environment, are a key interest in the research field. But talking from a biochemistry protein point of view, they're kind of also challenging to work with because of their low solubility. So our group personally does not focus on membrane proteins due to the difficulty to work with. But I would say, given that receptors are also involved in other disease settings, such as cancer, there's definitely a high interest in different receptors.
Great, thanks. In my questions, I've focused a fair amount on these other receptors besides ACE2, but it's fair to say it seems that this pair of studies has also yielded important new details about ACE2 itself, especially its interactions within the intracellular cytosolic tail region. What are some as yet other unanswered questions about ACE2 that might be worth asking in future research?
Yeah, so I've talked about like some of the SLIMs which we have assessed in the ACE2 tail, but there were still some that we did not cover from the prediction that Bálint made. So for example, there's the MPXF motif, which is proposed to bind to the PTB domains. And we could not find any significant binder for this. So this needs still further follow-up to determine whether the motif is functional. And the same goes for the binding motif for the IBAR domain. And as I stated at the end of my topic, this is a functional assessment based on affinity measurements, so that we still need to determine how the receptor uses the motifs, and then further on how SARS-CoV-2 infection uses these motifs in large extent or by extension.
Okay. Your work also points to SLIMs as levers to fight other pathogens, including Salmonella, HIV, and Ebola. Can you elaborate on this, and what might be some next steps along this path?
Here I can refer maybe to some work that we are actually doing in our group. We are doing something that is called proteomic peptide phage display. So you have peptide libraries and you screen for binding against different protein domains. And one of the libraries which we have consists of the viral proteome. So here we look whether the the viruses display short linear motifs to which human proteins can bind. So this may unravel how viruses, by showing these motifs, can hijack the cellular machinery. And the same also goes for the bacterial proteome. So that is kind of a high throughput technique to screen for viral SLIMs hijacking the human machinery.
Thank you so much, Bálint and Johanna, for these insights. To conclude, I'd like to ask each of you to provide a very brief, roughly 30-second parting thought that answers the question, what do you most hope for everyone to take away from today's discussion? We'll start with you, Bálint.
All right, thank you. So in conclusion, I would say our take-home message is that when we think about SARS-CoV-2 infection, ACE2 is an important receptor, but it's definitely not the only one. The virus can also hijack integrins and possibly other receptors to subvert the connected host processes, such as endocytosis and autophagy for its own needs. Identifying these receptors and the underlying pathways are crucially important to develop better host-directed therapies and to repurpose existing drugs.
Wonderful, thanks. And Johanna, over to you.
One key aspect to consider is that the function of the receptor is not limited to its extracellular, but extends to its intracellular part. We demonstrate here the functionality of various binding motifs in the ACE2 C-terminus, including an endocytic sorting signal. Furthermore, we can link integrin beta-3 tail to autophagy. We envision that cell-based studies will unravel whether the receptors use the cytoplasmic motifs in a biological setting.
Wonderful. Thanks so much, Johanna. This has been so insightful. I'd really like to thank our speakers once again. They'll be available in a follow-up Zoom room, again, as noted today, Monday, February 8th at 2 p.m. Eastern Time. And their papers are published online at the Science Signaling website. Again, thanks to our speakers and to all of you for attending this session of the AAAS annual meeting.